So I'll tell you a little bit about exactly what is available in terms of the actual nuts and bolts, the capabilities which you can then tap into as you want to develop your project. I think what makes this consortium unique is shown in this diagram here, at least for me. What we've got is AI UK providing expertise and money up front. I think their disease expertise is exceptional. They know a lot of people in the business, and I think that is very valuable, particularly if you want to bounce ideas off people. You've got MRCT providing the nuts and bolts resources to enable you to get your project off the ground. But I think what's really unique is having two pharma companies in from the start. You can see that Eisai and Lilly's involvement will be all the way through, hopefully growing as the data we get on your projects become more and more exciting. But having them on board right from day one, I think, is what makes this unique and is very valuable to this consortium. And I think that's a key point to stress. We at MRCT will provide the drug discovery framework to enable you to move your projects forward all the way through to the route to clinic. As I mentioned earlier on, we focused on small molecules and antibodies, but we wouldn't exclude other approaches. We have a big network of CROs who can provide other expertise. For example, if you have a peptide therapeutic that you want to make forward, or an intrabody or something, we can do that as well, but it would probably be in collaboration with another party if you don't have the expertise. So nothing is off the table. We'd like to look at everything that you've got that might be of value, and then we can take it from there in discussion with you as the best route forward. So for small molecule, we have 20 chemists at MRCT, which are potentially at disposal for the consortium. All our chemists have extensive pharma experience, so we speak the same language as Eisai and Lilly, which I think is great, because we will design compounds in collaboration with you, which Eisai and Lilly would like. We have well over 200 years experience between us in pharma companies, and have delivered probably around about 17 drugs between all the medicinal chemists. So we know what a drug looks like and can help you devise one the same. Supporting them, we have 25 biologists who are extremely experienced in both assay development and cellular pharmacology. If there's an assay you need developed to do a screen from, our group can do it. We have access to high-throughput screening robots and our own compound library. We have about 170,000 compounds which we can screen, made up of both diversity set but also targeted sets, for example, kinases, ion channels, GPCRs. And there's always the potential to maybe tap into what Eisai and Lilly might want to look at as well into the same screen if they are, if they have compound sets they think would be worthy of tapping into for the same target. We will do hit-to-lead chemistry with you. We can also do lead optimization chemistry, but I think at that stage we would certainly start to see excitement and involvement, more so from Eisai and Lilly, to help move things forward. And we would expect to pass the baton on, I think, at that kind of stage. For antibody drug discovery, we have 15 people experienced in antibody engineers. Over the last 20 years, we've humanized over 55 antibodies. Of which I mentioned 2 on the market, and a large number are still in clinical trials. So we've been very successful in that score, and we are developing new methods for humanizing antibodies beyond the straight CDR grafting, which we tend to use in the past, because that was MRC's method or modus operandi for antibody humanization. We will do biophysical characterization to make sure you've got an antibody which is suitable for treatment of either animals or humans, and we can do affinity maturation if the affinity isn't quite right. We can do that to improve things, and also to improve, hopefully, the biophysical characteristics along the way. So the resources available, we can also provide in silico modeling and screening if you don't have that capability in-house. We've got 2 computational modelers. We have access to a virtual library of many millions of compounds, which we can then screen and see what we can find. Very important if you've got structural information. We can also get structural information if you haven't, because we have a tie-up with Leicester University, who have an extremely good X-ray crystallography capability, and we've embedded a number of our staff within their X-ray facility there, and they can do X-ray and NMR structural information. So if you can't get it, we can from the route that I've mentioned. We'd also do ADME testing. For those of you who don't know what that means, it's absorption, distribution, metabolism, and elimination. So it's basically how a drug interacts with the human body and how it's broken down and excreted. And we'll do a lot of studies in vitro, which will help us understand what the stability of your compounds are that we've made together, and see which ones are the most drug-like, which ones are the most appropriate to use in the models that you want to look at, and which ones ultimately could form the basis of human targeted drug discovery. We have a network of CROs, as I mentioned, which can provide other capabilities which perhaps we don't have, and I mentioned things like peptide therapeutics. So if you want to go down that path, we know a person who can do that and can help you develop those things in the future. So I think just a brief word about what do we mean by drug-like. We often talk about we're looking for a drug-like compound. And I think Eric is right in the fact that a tool compound to prove a hypothesis doesn't necessarily need to be drug-like, so long as it's fit for purpose. If you're using it in an animal model, it needs to have the right exposure, it needs to hang around for long enough, it needs to have the right potency, and importantly, it needs to be selective enough that you can interpret the data. But beyond that, we're looking for compounds which are suitable for scale-up production. That's particularly important for antibodies, where you're looking for a expression in a cell line at a level that's useful on a production level. They could be administered to man, is what we're looking for, has the right profile, so they've got the right solubility, the right characteristics of oral absorption, bioavailability, and so on. Do they get to the right site of action? Otherwise, if they don't, they clearly won't work. Do they have the right potency at the target modulation that you require? We can help you with all of this to understand what's required and what's needed. And for small molecules, we would look for things generally which are Lipinski compliant, which have no toxic force, in other words, no groups that you don't want to see which might lead to toxic side effects, and they have good properties for absorption and distribution around the body. And for antibodies, we develop things together which get good affinity and good biophysical characteristics. So we're looking for something ideally which could lead to a springboard directly into a full-blown drug discovery program within AZ or Lilly. It gives them the best start, the best opportunity to develop a potential drug rather than having to go back to the drawing board and start again if we can provide them with good starting points. So ideally, the deliverables would be to provide our pharma partners, AZ and Lilly, with robust and collaborative projects which then can enter their hit-to-lead programs and beyond. We don't anticipate that we would just pass the project on at that stage. We definitely anticipate that you as academics would want to be involved in projects going forward, and you have that opportunity to speak to AZ and Lilly as we pass the baton on to see how you can be involved going forward. So we'll look at proof of concept and accepted models of disease is where we want to get to. That is debatable what those models are, but that's the advantage of having pharma partners on board from the start, because they can advise us in terms of which the models that they believe are the right things to prove efficacy in the clinic. Demonstration of target modulation, and we want to see an associated therapeutic effect. We want tools and reagents to feed into full-scale drug discovery program. Those tools and reagents will provide the proof of concept that we want to see to demonstrate that your target has good disease association and has a good chance of making a go of it in the clinic. There will, we believe, be opportunities for publication. We realise how important that is, and also there will be opportunities to look at funding for the development as the projects develop forward. If we get a great data package, that could form a very strong basis, for example, an application to the MRC's Developmental Pathway Funding Scheme or the Wellcome Trust to get additional money to help develop the projects further. Indeed, there could also be money from sources within the consortium who might like to develop projects further. So these are things that could be the upshot of what you do. So what we really want to see overall is seeing your basic research translated towards the clinic. And I hope you're here because you want to do the same. We ultimately want to see significant patient benefit, and we hope that some of the ideas that you have in this room now might ultimately lead to exciting new developments in treatment of dementia. Ultimately, if that happens, all the parties within the consortium, including the academics, will share in the revenue from successful projects. So there is a payback if your project is successful, that you would expect money to come back to you and your institution to help drive further research in the future. Ultimately, we believe this consortium is set up so that everybody wins. We believe that the academics will win by virtue of seeing their projects progressed and hopefully getting money back from successful projects. ARUK and MRCT will win because we have the opportunity to get involved in these projects, and we believe that ASI and Lilly will win because they will get a good pipeline of strong projects coming forward. But overall, we believe that the patients will win because we believe that this will deliver some exciting new therapies which will have significant impact on symptoms or even disease progression of patients who really need that. I think with that, I'll pass on to Duncan, who will tell you a little bit about the application process and how it will all work.